So I would like to express my gratitude and thanks to Dr. Keshav Singh and Dr. Thangaraj for inviting me here to this fascinating workshop on human diversity and health disparities. So my job here today is to give you some idea of what are the bases of disparity from the immunological perspective and what we know as of today and where should we go from here. So cancer poses a global health problem. We know that in next decade we will have 21 more million cancer cases globally, and that would put us onto a burden of almost $500 billion. So this is a huge priority as we go along in this decade. Now, if you think that cancer is a modern disease, that's not the fact. First recorded case of cancer is Cleopatra, and our anthropological ancestor Australopithecus bone showed a tumor formation which is 1.9 million year old, and we also know that plants have tumors. So cancer basically is a consequence of multicellularity. It has evolved along with us. The cancer development also does not happen quickly. It takes a lot of time, long time. So in any cancer development process, the early stages are always reversible, but when they reach to the later stages, they become carcinoma, and when they become invasive, they become lethal. This is where it's very hard to treat a patient. We can treat a patient somewhere here. So for many different types of solid cancers, if you look at this profile, for colon cancer, it would take at least up to 20 years for that cancer to become lethal, going into clinically pathological stage. For head and neck cancer, again about 10 years. For cervical cancers, it would take about 13 years. For lung cancer, the non-smokers or smokers would take about 20 to 40 years. So and so forth. For breast cancer, prostate cancer, again a very long period of time is needed for that cancer to become clinically pathological. So behind cancer development, there is a strong interplay between cancer and immune cells. This we know for a fact today, but there are a lot of components behind this scheme of interplay. So the focus that we are going to have today is on lymphocytes. Now we know that there are 10 different types of lymphocytes known as of today. So on the left side we have T lymphocytes. On the right side we have innate lymphocytes. Earlier we used to call them NK cells. Those are cytotoxic ILCs, innate lymphoid cells, but we know many more now. Now, the striking feature about these adaptive lymphocytes as we know them is that they act against specific molecular patterns. We call them epitopes, and they could be of peptides, polysaccharides, or glycolipids, and we call them antigens. And those antigens are recognized by lymphocyte receptors, which are highly variable due to the nature of somatic VDJ recombination. And this recombination gives rise to a repertoire of about 10 billion cells. All of us here have 10 billion lymphocytes, and in Indian system this is 1 kharab. So about 20 years ago, we didn't even know that lymphocytes can react to tumors. And to address that question whether lymphocytes can react to tumors, we needed identification of cancer antigens. So these were the original papers in 1991 from the group of Thierry Boone, where cancer germline cells antigens were identified. And they also had identified a mouse model for those antigens, P1A, and many of the human tumor antigens that we know today, MAGE, BAGE, GAGE, NYES, all belong to this category. So I became part of this European community project between France, Belgium, and Netherlands, where we developed some TCR transgenic mice on different backgrounds. So all CD8 T cells in these RAG knockout backgrounds were specific to P1A epitope, whereas in these backgrounds, the majority of cells, about 40% of the CD8 T cells, were reactive to this P1A epitope. So using these mice, we addressed three questions: whether T cells develop in thymus against tumor antigens, or if they develop, then are there same number of CD8 T cells in those different strains of mice, and whether those T cells can recognize tumor and kill tumors. So the answer to the first question was published in this paper, where we showed that these two are different backgrounds of mice. You can see that in beta and D2 background, we have 85% of the CD8 T cells reactive to P1, specific to P1A. In the DBA2 background, we have 62%. Now, if we cross this beta and D2 onto DBA2 for 4 generations, the F1 has about 81%. In terms of percentage, 85% reducing to 81% may not seem to be very significant, but the lymph node size in these backgrounds are smaller compared to beta and D2, and that brings the absolute number very much significantly down. And this is a positive control of RAG knockout transgenic background, where we have almost all cells reactive to P1A. Now, the question where we asked whether T cells react to tumors was answered in these two publications. So we found that T cells can recognize those tumor cells that express P1A, and they can reject tumor. In addition, what we found that those T cells can also make a functional network with NK cells. And so it's a network of T cells and NK cells that ultimately rejects tumor. So these were the landmark studies where we showed that T cells against tumor develop in the body, and they can control tumor development. And building on these studies, later on many other groups then confirmed, and now we have a cycle called cancer immunity cycle, very well established. And by cancer immunity cycle, what we mean is that at the site of tumor, release of tumor antigens in an interferon gamma laden environment, mature dendritic cells can take up those antigens, get matured, and then migrate to the lymph node, where they present that antigen to a T cell. And those T cells then become cytotoxic, migrate back to the site of tumor, and kill the tumor cells. Now, the activation of T cells, I would like to remind you, requires 3 signals. Signal 1 is called antigen recognition signal, which is the binding of MSC peptide on an antigen presenting cell to a T cell. Signal 2 is a co-stimulatory signal, where co-stimulatory molecules on dendritic cells further activate or co-stimulate TCR signal. The third signal is called checkpoint signal, which becomes activated later in the cycle of T cell when the T cell function has to contract, or when T cells become exhausted after they have performed their function. So, in a conventional sense, this is how T cells can reject tumors. And this appears to be very simple. If the life is like this, none of us would have cancer. But the thing is way more complex. What happens if you analyze, so here in this case we are analyzing 2 patients. One patient has infiltrating CD8 T cells at the site of tumor. We also have high expression of PD-L1 in that environment on stromal cells or tumor cells. I told you the third signal, checkpoint signal, so PD-L1 is part of that third signal, which tries to inhibit T cell function or exhaust them or bring their function down. The other patient does not have T cells infiltrating into the tumor and does not have those molecules. So these 2 patients pose 2 different environments. One environment of patient 1 is called T cell inflamed tumor environment, and the environment of patient 2 is a non-T cell inflamed tumor microenvironment. They both have very distinct characteristics. So for a T cell inflamed tumor microenvironment, what would happen is that at the site of tumor, there will be upregulation of PD-L1, the checkpoint molecule, which would suppress T cell function. There would also be upregulation of IDO, which is an enzyme for tryptophan metabolism, which takes away nutrients from T cells and thereby suppress them. There would be also loss of antigen presentation, and they also bring in a cell type called T regulatory cell, which is also a suppressive cell for cytotoxic T cells. So these tumors in this type of environment escape by suppressing T cell function. The other type of tumor, which is non-T cell inflaming, has other problems. For example, at the site of tumor, there would not be any innate priming. So no antigen presentation, no T cell function, so lack of T cell recruitment, and they have upregulation of oncogenic signals, which then keeps the tumor thriving. So tumors grow here as a result of malfunctioning cancer immunity cycle. So essentially, so T cell function is only one component behind tumor surveillance and tumor rejection. There are more factors. For example, environmental factors, the immune diversity, including T cell diversity, and the somatic mutations of cancer. So the picture is even more complex. And it's an interplay of this immune diversity and the environmental factors combined with the tumor heterogeneity due to somatic mutations. All those together make the problem further more complex. So our hypothesis is, and I apologize for this typo, so our hypothesis is that ethnic immunogenetic differences among various populations on this earth would affect lymphocyte repertoire, as is true in this hall. For example, each one of us has a different set of T cells in our body, T cells and NK cells, and that puts us differently into how we will respond to cancer and how we would respond to therapy. So essentially we will have a different set of lymphocyte repertoire. That means we will have a different anti-tumor immune surveillance, and thus we would have different therapeutic outcomes. So this hypothesis is logical on the basis of the known fact that our MSC and receptor complexes, they all have vast variety of polymorphism. They belong to the most polymorphic regions in the immune genome. So we need to study this much deeper, and there has been lately, 2 years ago, this paper where they tried to understand the immune landscape of cancer. This is a systems biology paper where they analyzed all tumors that are available in TCGA data sets. So there are 33 cancer types available in this data set, and they profiled all those tumors according to immune phenotypes, subtypes, and they call them C1 to C6. So one immune subtype is wound healing, interferon gamma dominant subtype, inflammatory subtype, lymphocyte depleted subtype, immunologically quiet subtype, and TGF beta dominant subtype. They went on to further analyze those on the basis of different ethnicities. So they made 4 different distinct clusters. The AC1 cluster is the East Asian cluster, AC2 is a mixed European cluster including Native Americans, AC4 is the predominantly African ancestry, and AC3 is the European ancestry. What they found is, taking an example of this one bladder cancer, we can see that lymphocyte populations in all these 4 different ethnic groups vary in their number, in their proportion. So like East Asians have the lower number of lymphocyte fractions. If you look at the PD-L1 expression, there are also variations. So they compared the 4 ethnicities, and we can see that those variations changes, and in AC2 we have slightly higher proportions. They also found that females compared to males also have differences in the PD-L1 expression. PD-L1, remember, is a checkpoint molecule which suppresses T cell function. In this figure they showed, they compared other ethnicities, and they found that if you analyze these components of immune cells or PD-L1 expression, they have a diverse range of expression. And on the bottom, they are the different cancer types, including lung cancer here. I would elaborate a little bit on lung cancer now on and show some of our own data that we have started working in this direction. So we know that in Africans, blacks, we have high incidence of lung cancer, and that is irrespective of smoking. Also, women in Caucasians and blacks also have high lung cancer. So we noted in 2011 that bone marrow cells from lung cancer patients had lower expression of DLL1. DLL1 is a delta-like ligand belonging to the Notch signaling. And this is downstream signal molecule HES1, which was also downregulated. We digged into TCGA data sets and found that there also we have a similar profile for N2 Notch receptor 2 and protein. Now, as I just said, Notch is a very conserved signaling, and it has 4 receptors, conventional receptors, 5 Notch ligands, and 3 canonical target genes, but it also intersects with many other signaling pathways in the cell, including PI3 kinase, AKT, and many others that ultimately affect cell cycle progression, apoptosis, and transcription factors of many signaling pathways. So any changes in any component of Notch would affect many things in a cell. Now, in terms of immune development, Notch also plays a significant role. So, for example, the development of Th1 or cytotoxic T cells relies heavily on high expression of DLL1 on dendritic cells. The differentiation of Th2 cells would require low expression of DLL1 and 4 and high expression of JAK2. Same way for Treg, we require low expression of DLL1 and high expression of JAK1. So we built this working hypothesis that if we can promote somehow DLL1 signaling in T cells, we can improve their function, and if we can suppress JAK1 signaling, then we can overcome the immunosuppressive signaling in the tumor microenvironment. Because JAK1 was involved in the Treg differentiation, which are immunosuppressive, and DLL1 is involved in the differentiation of effector T cells. So what we did was we engineered some Notch ligands. So multivalent delta-like ligand 1 and another monovalent jagged 1. And so co-cultured these ligands in knife, along with naive T cells in an in vitro stimulation culture. We can see that based on these CD44 and CD62L molecules, which are markers of T cell activation and memory, we see that the red numbers here are the T cell effector memory cells, whereas the blue numbers depict T cell central memory cells. And if you look more further here, the T cell effector memory cells, in the presence of clustered DLL1, reduce the expression of PD1. And that means that those T cells now can function for a longer period in the activated state. So this was a good thing, and then we also analyzed freshly resected lung cancer patient samples in collaboration with Pierre Martin at Vanderbilt, and those 10 patients showed us that JAK1 expression on dendritic cells highly correlate with PD-1 expression on effector memory cells. We also noted that DLL1 expression on dendritic cells also correlates with various Notch receptors on effector memory T cells. In lung adenocarcinoma, we also noted from TCGA data sets that expression of JAK1 positively correlates with PDL1 expression, whereas DLL1 somewhat negatively correlates. And for the PD1, there was no significant correlations. So for the DLL1 high expressors, we found that the survival is longer. So high expression of DLL1 correlates with longer survival of patients, whereas the high expression of JAK1 correlates with poor survival. Now, stratified with different ethnic groups, we see same patterns, that high expression of DLL1 correlates always with longer survival of the patient, whereas high expression of JAK1 always correlates with poor survival of the patients in all ethnicities, including Asians, although our sample size in Asians is very low. So based on all these data that I have shown you, I think I have convinced you that we need an elaborate Notch-based signature which can predict whether the patient would be susceptible to cancer in the first place or would respond better to any type of immunotherapy. And then, same way, we need to go on to perform a comprehensive profiling of immune signature so that we can inform and use that for better therapeutic purposes. So, this is my last slide. So, I would also like to emphasize that the biology and the genetics only contributes to about 20% to the diversity. The rest 20% comes from the environment, but 50% comes from our own behavior, and which is not trivial. So from a Cancer Research UK data sets, obesity predisposes us to 13 different types of cancer. So I would like to make a point that we should not ignore the 3 key essentials of our lifestyle. We should always try to eat healthy. That means having good microbiome, which I did not touch in this, but microbiome has very significant impact on immune response. We should keep moving more because, as Dr. Keshav Singh would emphasize, that we need to have better mitochondria, and that helps to have more mitochondria. And I did not show that story today, but we also know that mitochondrial signaling and mitochondrial mass are important for immune networking, for better function. And the third component, we all should laugh freely and be happy. That also has scientific basis because all lymphocytes express neuroreceptors. So the more happier you are, the better your lymphocytes are. So finally, these are the people who contributed to some of the work. The upper line is all graduate students. These are the current students. All these are postdocs in the lab, and these are the current members. This is a young faculty in the lab, and this is a recent technician in the lab. And of course, in addition to the lab, we have several collaborators who help us in various ways, and the funding, and of course, if you want to contact me, you can always reach me.
Thank you, Dr. Anil.
Thank you.
The talk is open for discussion. We say that